laboratory materials. Now, we're going to be beginning the lab part. I'm hoping that the kits will start to show up this week, and so I'm going to be focusing on things you won't need the kit for, kind of steering it around that, and then we'll eventually, hopefully, when I get about at least 90%, and then we'll care about working with those that don't have it yet. I have a kit they sent me, and so if push comes to shove, I can mail that to somebody if they need it. Although I need it to use the demo, but I can talk a lot through it. We'll see how it goes. So there is a page of a document that has all sorts of Latin terminology, and some of you may already be aware of these things. This one is Latin terminology for microbiologists, so it shows a lot of the words that are associated with microbiology, and there are Latin terms. And so, as I say, it's all part of my philosophy of what you want to do is to learn the material, not memorize it. And one way to help is making it related to Latin, and so you're learning it, and you won't forget it, and that's the key. And these are the terms that you'll be using in your career. I also have microbiology made ridiculously simple. It's not the best illustrated book in the world, but it's there, and it's always nice to get a different kind of viewpoint on some of the materials if you're having trouble reading that. And again, I offer that as a suggestion whenever you're going into your upper level courses. You're going to be taking some courses that require a lot of additional type of insight maybe than what the textbook is giving you. So always try to find an alternative resource even if it's from the library. I have the OpenStax Microbiology. I would very much like to use that book, but it's already being utilized by the 275 class, and we're not allowed to use it. It saves a lot of money, and it's actually very well done. So if you get a chance to look at that, it's a great resource. So tools of the laboratory. And again, this will follow and integrate as best I can with the eScience labs and the kit. And this is sort of the components. Now, we had the abbreviated kit because the faculty tried to, we sat down for hours to figure out what are the basics that we need to cover to try to save money. The full kit with everything in it's a lot more than what you had to pay, and we didn't think it was necessary, so we got the bare essence. But the things that I want to focus on is that aseptic technique, and I'm going to work hard to do that, how to use the microscope, and I'm working on that today, and you'll have aspects of that showing up today. So I will provide a sort of a video on how it's going to be organized. I'm trying to figure out the best way to do this. There is no set standard for this. I've never had to go through this before, but I got you covered. I've been doing microbiology for a long time. I taught at the vet school way back when, and I had to use some really interesting types of techniques to work with the veterinary students not being in the field. So I think I can pull on some of those experiences. Even though that we're not in our laboratory setting, the normal one, you still have to wear the safety glasses and the like. I tell you, I had an experience when I was an undergraduate at NC State that another team, not associated with what work I was doing, they took a lunch break. My partner and I were doing our work. We usually didn't have the luxury of doing lunch because of our class schedules. Well, anyhow, they left their distillation going, and it exploded right in our faces. I was wearing goggles, and there was shards of glass on my face and things, but not in my eyes. My lab partner wasn't so lucky. He didn't think, he said he looked goofy in a face mask, and now he lost sight in one of his eyes. And so safety glasses are just one of those things that I take very seriously. The lab coats, it should be part of your kit. And obviously we use the cloth type or the paper type, not the cloth type at the school, because they are disposable, and cloth tends to retain various types of things, and these other types of fabrics tend not to. It's just the composition and that sort of thing. But the longer the coat, you don't want to wear flip flops. You might drop things on your feet. I really would not wear shorts. I would wear regular pants or whatever, uh, just as long as it's full length. That way, if you spill something, it won't get on your skin right away and that sort of thing. And those are just safety features. So in the lab, of course, we do the best we can. We have various pieces of equipment that I'd love to have shown you, but I'm hoping that through this chapter 2 you'll get an understanding. I will prepare various different types of presentations, so there's a little bit more of the maybe videos to watch and that sort of thing, and I will pull together some additional videos that are supplementary. I've collected some that I think you'll like. They're optional. They're only there if you want to review or have something. But all of this will have as documented of what you need, so not to worry. Some of you have already sent me some of the chapters or the units for the lab, and that's fine. But my goal is to go over all of those labs video wise, so you'll have those, and we can work through those together. So in the lab, one of the things that we do is that we have to manipulate and visualize bacteria. Now, the concept that I try to convey to you, what you're looking at on this plate, is a mixture of bacteria and fungus. There's fungi of various different types, and each of which you can't see with the naked eye. But as they grow and they start piling up the cells, you can obviously see them. So a colony is really a growth of a single cell, because they don't have to have male, female, and all that. It's they divide by binary fission, and they replicate every 20 minutes, some of them, and we start to see so many cells that we can actually visualize it on a plate. Now the plate is made of agar, not agar, but agar, and it's made from seaweed. It's you can get different refinements of agar, but the typical agar is a seaweed that's ground up and it's sort of translucent. But that itself, in itself, is not the media in which to grow the bacteria. It's just the substrate. They used to use a long time ago, before Petri plates and agar, they used slice potatoes. Of course, internally the potatoes would be sterile, and then you could grow bacteria on the surface, and there's enough sugars and various things in the potato. But we don't do it that way anymore, and so the agar plate's what we use, and we have to add things to the agar plate, the agar mix, and which feeds. It gives the sustenance to the uh, microbes that are there, and we're going to talk about some of the nutritional needs and things which are important. I worked out minimal media for lots of various different types of organisms. Minimal media means is the minimum of requirements for these organisms to grow. So I had to identify all the various pieces and parts. I knew every single chemical that was in the composition and that they could grow from. And it would take me years to develop a minimal media for Treponema pallidum is one that I worked on. It was really, really tough, but it worked out. It worked out. Uh, so what we use the media for, though, is that we can characterize, visually look at the bacteria, and it tells us things. It either grows. So the whole aspect of the lab that we're going to be talking about is things that we add to the media that helps identify. In other words, the bacteria, all they have to do is grow and respond, and when they do, it tells us something about them. It gives us the characteristics about them that helps us to identify them and understand the properties. So, given though it is an artificial world that they're growing on, not the host tissues and various things, so we're getting sort of a subset, a false picture of what might be actually happening. But we can put antibiotics in the plates if we want, and then they have resistance or all sorts of things that we've learned. Now, the pathogens, once they get in our blood and system, you can see that we have some really ugly things going on here. And the skin actually is the largest organ system, but it's a tattletale for a lot of these types of infection. Now, you can see we got a bulla, some sort of a burn perhaps, or it could be even fasciitis, where you see the skin is, you would probably take a magic marker and draw around the perimeters and see what's going on. And also you would find out from the patient what happened and those sorts of things. But it's up to us to use clinical means to identify these. And some of the lesions are characteristic. It could be due to various types of pathogens like staph. This one over here, I believe, is staph. This could be tattletale of treponema pallidum or syphilis, and you start to see these little lesions showing up, and then you'd have to look at sexual organs and things like that to determine if it has the cankers and all that stuff that are characteristic, and you can re-isolate the organism, and we'll talk about that. And then a lot of times we see viral diseases that give us a sequelae of the skin, and helps us to identify, is this one type or another of a type of virus that infects, like chickenpox or whatever. And then using blood samples to try to identify these things. So understanding how organisms look like on a plate. Obviously, you see a variety of different organisms here, yellow ones, white ones, all sorts of things, and they're growing together. Our job is to get these in pure culture. So when we streak plates, we have to streak for single colony isolates. Now, you're going to get tired of me talking about that, but it's really, really important. And I've got some techniques that I want you to learn that doesn't even require using plates or the whole 9 yards, just to start to practice and knowing what to look for. So I would take a single colony and restreak each one of these to grow them in pure culture. And then if it were, this could be perhaps a sampling from your hand or something. You can see hairs and things. It could be environmental sampling. And you'll find that the microorganisms around us are ubiquitous. They're everywhere. And that's part of the issue, of course, that we want to try to keep our environment, even though we can't see them until we grow them. So one of the experiments that we're going to do is I'm going to have you put your hands or something around your house and see what we grow. But because it could be potential pathogens, you can see a piece of tape holding the plate. We always tape our plates in case you drop them or something. We don't want to create an aerosol, and because these are unknowns in high numbers, we want to be careful. So even though they're around your home and that sort of thing, it still is potentially dangerous or hazardous. And I always provide a list of organisms that we work with in the lab, although this won't be appropriate so much for us. But I'll tell you, I can look at these colonies and I can see Staphylococcus aureus. I can see perhaps Klebsiella, and a lot of other potential pathogens here. So even a salmonella, perhaps. I'm not sure. But the idea is that there's high numbers, and we can't let our guards down. You never let your guards down. You have to assume the worst and hope for the best. And so we just take the precautions as a microbiologist, and I'll share those as we go along. We see various different types. This is a fungal, the fungi that are growing on here, and sometimes they do look like bacteria, but when you go to look at them under a microscope, that gives you that next level of envisionment that tells you, or information that helps you identify these. And again, it's important to do the gram stains and various things along the way, even though that, well, I think I know what it is. You can assume. Every now and then, if you don't do it by the book or by the numbers, you can get burned by that. So it's best to always do the work. And we go. Now we're going to talk about, and there's a section that I asked you, or it's in the paperwork when I go over it, that I'll ask you to go and look into the four different layers of the biological safety levels. There's four. Biological safety level 1. The lab that we typically use at the school is a BSL 2, and there's certain organisms that are potential pathogens that we take additional considerations, and I will show you that. This looks like to me like a BSL 4, and there's certain things. It could be a 3. There's just not quite enough information here. It says at the highest level, so that'd be a 4. But if you look around, everything that's in the lab stays in the lab. It never leaves the lab. And the suits are positive pressure. You can see the air hoses that they carry around is positive, so organisms can't get in. They're just kind of blown out away from you, and that's what you want. And then there's all sorts of dating and information about the age of the suit and all that that has to be with you at all times. And then getting into the suit has a rigmarole. You take your clothes, and then you put on scrubs after you take a hot shower and that sort of thing, and scrub, and then you get into your suit and you work all day, and then you reverse the process, and when you leave, you should be as clean as a whistle, and not going to transmit anything to outside of the lab or to your family. So containment levels are based on organisms not contributing to human disease, to or we don't really know. Sometimes we just use a containment level to step up just standard safety precautions and sterile technique. Microbes that cause mild human diseases, that's the BSL2. That's what we run at Wake Tech. And there's all sorts of standard operating procedures. And one of the things, and it's a pain, but all your notes and things that you might take in the lab stays in the lab. And then I take a precaution and a microwave following a procedure to sterilize the paperwork, and I can give it back to you later. And it's what we've done, and it works. And it's just a protocol that we use to try to minimize any potential, or try to eliminate the possibility of getting contaminated yourself or something else. We can't use cell phones in the lab, those sorts of things, and you have to gown out, glove out, and wear goggles depending in the laboratory setting. Now, 3 is serious and lethal human diseases. 4 is severe fatal human disease with no cure. So this would be your Ebolas and AIDS and COVID-19, that sort of thing, as far as we know, as it stands. So that's the overall standards that we use for BSL. And of course, we have all sorts of different PPEs and things that we wear, and techniques that vary from location to location. Hospitals have their own SOPs, so always ask wherever you go for a written copy of SOPs for when there is an outbreak, so that you're aware of them, because you'll never know when that time comes, and you'll be prepared. You'll know what to do and how to do it. So that's one of the things that I always kind of focus on, is I want you prepared, even though it's not a situation. Let's say COVID-19 doesn't exist and all that. I want you already know to know how to put on a PPE, how do you put on gloves properly, and that sort of thing. And it's easy to do. There's actually sometimes hospitals will have courses that you can take, or classes for that. I know at NC State in the biotech, the BTech Center, you can take courses. It's set for pharmaceutical applications, but they have high standards for the clean rooms and things like that. What a great experience! And a lot of times you don't even have to be an NC State student to take some of those courses. It's nominal fee to do it. I would highly recommend you get the experience. It won't hurt. So microorganisms. This is under an electron microscope. We're going to talk about that. And electron microscopes are typically just grayscale, but you can colorize certain features post processed. So a certain gray level, a level of grayscale, will get a color, and you can just do that programmatically on a computer. Anyhow, you can pull out features, so you can see red blood cells and various microorganisms, and it really does kind of show you that world. It reveals the contamination of what we talk about portals. If you remember, in chapter 11 we talk about exit portals and entry portals for various things. This is what we mean. This is what we see at the microscopic level. And the microorganisms that we might look at, these are bacillus type. This is most likely a pneumococcus or a diplococcus of some kind. Here's a virus. This is under electron microscope. And this is typically grayscale what we see. I'm not sure, that looks like maybe a Zika virus. But you can see the detail and how these microorganisms, the structures that they have at that level. It's important. Here's HIV. It's a model of which you can see, and a section. You can do that with models. You can take a section of it out and you can see the capsid on the inside. This outer part is what interacts with the human. See, it can't replicate or do anything without a host, and all of this implementation has been encoded by the RNA that's inside this capsid, inside that, where the orange part is here, and inside that is RNA. That gives all the instructions for making all of these pieces and parts. So you're going to make that. You're the host that's been pirated, and then it assembles, and then out it goes as a full-fledged virus, and not only one but billions and billions of these things. And so you can see the intricate structure of this. This is a representative. Of course, it's not the actual virus, but it's based on what we know about. And I actually printed one of these. It looks pretty cool. So we'll explain the 5 eyes, but as far as a lab is concerned, so you saw in chapter 1 and that some aspects of the 5 eyes. Discuss 3 physical states of media and compare and contrast selective and differential types, and defined and complex. So I've already kind of mentioned these things, but we'll go over these. So in the folder for 2, I have the 5 eyes listed as a graphic, just very graphic. If you'd like to have that, it's there. So inoculation, that's a sample is placed in a container of growth media via inoculation, so we're adding the bacteria to it, the inocula. And the media could be solid or liquid or a live animal such as a chicken embryo. Incubate it, or incubation, as we put it in the growth conditions that are optimal. So here is a incubator. A lot of times the moisture is kept at a certain level, and let's say 37 degrees or 42 degrees centigrade, whatever is optimal for the organism. Isolation, once the cultures have been grown, they may need to be re-inoculated and incubated and such to separate. So here is single colony isolate here. This is spreading the bacteria in such a way that we can dilute it, and you can see the dilution over here, and we're getting the single colony isolates. The reason why this is so important, that each of these colonies represents a clone. And so I might mark on the back side of the plate that I took from this colony here, and if I'm doing research, then I can come back to this plate and always get the same clone of that bacteria. It's just one of many things that you can do with single colony isolates. Also, it gives you a good indication that your culture is still pure, there's no contaminant in it. Or if we're looking to some characteristic of how that organism's interacting with the media, having a single colony isolate helps us to detect that. Inspection: colonies on the agar or broth cultures are observed macroscopically and microscopically for possible understanding of what the organism looks like, how we classify it, how we bin it, and those things are important. And we're doing that. And identification we can use at the clone level. In other words, taking a colony or portion of the colony, we can now run some genetics or do additional types of tests or immunological tests or whatever on that, and we've identified some characteristic. I can go back to that colony and then reproduce the exact work. Another clone may not be the same. Sometimes they can have mutations or they lose a certain characteristic. So that's why it's important to work with the same colony every time when you're doing research or whatever. So the inoculation part, we culture to grow microorganisms. The medium, now this is not somebody who's psychic. We're talking about media. Media is different to be singular and plural. Nutrients for the growth of microbes, and I can tell you that I've fed more than McDonald's, hands down. I can't tell you how many microorganisms I've grown in my career. So you can see this person, by the way, is inoculating a plate. There's certain various types of techniques, and I'll show you some, but they have their lab coat and their gloves, and they're trying to treat, I would assume that there's a Bunsen burner nearby, and they're using aseptic technique. The lid is over the inoculation loop so that you minimize any chances of contamination. So when you inoculate, you introduce the inoculum into the media to culture the microbes, and that's important because how you do that allows you to get your single colonies and that sort of thing. Now we can get these cultures directly from clinical specimens, from urine, let's say, or anatomical sites or whatever, and spread it on the media. And we may have to do several passes to make sure that we have single colonies, but that's what we need to do. Incubation is just once we have some clue of what organism that we're dealing with, then we can choose the right temperature at which to propagate or to grow them. And sometimes they may be sensitive to oxygen, so in those incubators we can have gases in there that devoid the oxygen, and so we have carbon dioxide, and that allows certain organisms, anaerobic ones, to grow. So understanding what we're doing and how we're doing it is key to being a microbiologist. So pure culture is what we want. A lot of times we talked about having uh, uh, multiple organisms causing a disease, so we don't really know, so we have to isolate them and purify them. So you can see this mixed culture on a plate. You can see the yellow and the white, and our job would be to isolate, restreak each of these so that we get a pure culture of each. But doing a single colony isolate shows you that you in fact have a mixed colony or a contaminant. Now, it could be the same organism, but it's lost or it loses an unstable characteristic. That's always a possibility, and that would require more work. And here you can see a contaminated culture where the predominant feature is this sort of orangish colony, and then all of a sudden you see this white, and that would require, I would go back and just isolate a single colony from that. And so can be determined if they're contaminated or not simply by restreaking them on a plate, and we can look at the characteristics. So we can use culture media that can be test tubes, flask, or petri dishes. And the flask, we could have liquid media. Test tubes are smaller, more manageable. We can apply aseptic technique a lot easier using test tubes and that sort of thing. Media may be inoculated with loops, needles, pipettes, swabs. Most common is the microbiology the microbiologist loop, and you can see that little ring at the end and that nice little handle on it, because when you heat the rod, if I've been at bench working all day, this thing starts to glow up here. It gets quite hot. When this tip gets worn out, and sometimes we have to put a new loop on there, they're made out of platinum or something, you can clip the end off. Now you have a needle, and the needle you can use to inoculate certain types of media to see if they are motile, or we'll talk about how bacteria move and that sort of thing. And pipettes, we never mouth pipette in the lab, but we use pipettes to move various segments of liquid, and then swabs used very frequently to swab perhaps a patient or a source of something that we can use now to inoculate a plate. But always in a microbiology lab, we take sterile technique is absolutely mandatory and has to be practiced. The better you are at it, the more less likely a chances of having something contaminated. Three physical states. We have the liquid state, the semi-solid state, a solid that can be converted to a liquid, and a solid that cannot be liquefied. So some of these depends on we want to grow things in a liquid, or if we want to look at how motile, or if something uses flagella that the bacteria actually move, we use a semi-solid media, so you can actually detect movement. You can see it. A solid that can be converted. Sometimes we want to know if a certain enzyme liquefies a media. It lets us know a certain characteristic, or most of the time we want to manipulate bacteria that we don't want the media to be liquefied. So the agar is complex polysaccharide that's been isolated from Delidium, and that's a seaweed. And we also know this as a thickener that's used in ice cream and various things. Similar, it's not ideally the same, but unfortunately it is somewhat toxic, even though they put it in foods. But don't get me started. Solid at room temperature. So it's sort of like Jello from the standpoint, if you heat it, it becomes molten. You allow it to cool, it congeals. And so it's a solid at room temperature. So there's a temperature at which it will liquefy, and agars around 100 degrees C and solidifies around 42. So let's say I want to make a blood agar plate, so I'd add various types of media in the agar, and then I'd cool it down to about 75 degrees or so, and then add the blood, which blood's sterile for most practical purposes, and I would add it to the agar, mix it, and then I could pour the plates. So we take advantage of our understanding of temperatures. Now, anything above 70 some degrees, 75 or so, will lyse red blood cells, and that's no longer blood agar plates. That becomes a chocolate agar plate. I guess somebody made the mistake, justified it. Oh, we can use that for, in fact you can use that for growing other organisms that require some unusual components in the media. So, but typically we understand the temperatures. So blood agar and brown, having different types of agars, I guess we could say, knowing temperatures at which it's safe to add these components. Agar is flexible and moldable, and you'll see that this semester. It's really, I have you kind of explore it so you're familiar with it. And it's not a digestible nutrient for most organisms. There are some fungi that will eat the regular agar because it's a beta-linked polysaccharide. It's a plant. Some bacteria can, of course, break that down, and we have to put a higher concentration of agar in there. So you can see various different types of organisms that the fungi growing on agar plates, and sometimes these look like minimal media type of agar, or maybe a type of agar that doesn't have a whole lot of nutrients in it, but these organisms can grow. And you can see how they utilize the media, and sometimes they add color components because of their byproducts or whatever. So we see some bright colors at times. This looks like bread penicillium, where penicillin came from. We'll talk about how that happened. And so it's interesting. These are out there. So there's your liquid, your semi-solid, and then you can look at this is stab for looking at motility of bacteria, and you can see it's not motile. This one's motile because the whole thing, it looks like it kind of swarmed everywhere. So that semi-solid is used for lots of different things, but that's one of them. And then sometimes we make a solid and we make a slant. In other words, it was heated and then cooled at a angle, and there's reasons that we do that. That makes a larger surface in which to grow bacteria, and we can stab the area, so you can have surface and a stab in one tube, and it just makes very flexible in terms of trying to identify and manipulate bacteria. Defined and synthetic. So I would highlight this. This is really one of the meats and potatoes of micro. Defined or synthetic is the composition is precisely, and I would underline this word right here, precisely. We know everything that's in there. Obviously, it goes without saying that the water that we use is either distilled or double distilled water. That way we throw out variables, things that might be in the water that for whatever reason the microorganisms might be susceptible to or whatever. It's really important. So when we say defined, we want to know exactly everything that's in there. And so we would add sodium chloride and nicotinamide, and it just goes on and on and on, various things that we'd have to add in order for the organism to actually utilize a composition that we know exactly what it is. It contain pure organic or inorganic compounds that vary a little from source to another. Molecular content specified by means of an exact formula. So we know everything in a defined or synthetic media. So there is no doubt. And so complex is that one or more components are not chemically defined, like brain, heart, and fusion. Now, I couldn't tell you what's in brains and hearts and various things, but a bunch of stuff. And that's for complex, that's good enough. I mean, bacteria, some of them love, E. coli, love brain heart infusion, BHI plates, love it. Nutrient augurs and things like that, which can contain extracts of animals, plants, and yeast all kind of mixed in. Or you could have blood auger plates or serum and meat extracts or infusions and various different things that certain bacteria just love to have. You notice it doesn't have any ice cream media or cotton candy media because, well, microorganisms are really not into those sorts of things, I guess. So here's some defined media for growth and maintenance of a pathogen, a Staphylococcus aureus. And so you need 0.25 grams in a liter. It's assumed unless it tells you otherwise. So it's dissolved in a liter, which is 1,000 mLs or milliliters. We're going to talk about units if you're not familiar with those. I have a supplement called Solutions and Dilutions that should help you with that. It's there's going to be other courses that you're going to have to take in nursing, and it depends on what schools you go to as to what they call it, but it's really more of a pharmacy type of an arrangement. So the pharmacy schools and things like that usually help teach nursing, and so you're going to have to learn some of the standards and how to do this. And I'll help you with some in the micro because we use a lot of measurements and various things. So cysteine, histidine, leucine, these are all amino acids, and you need 0.25 grams in a liter of distilled water, buffered to a pH adjusted to 7.0. 0.5 grams of these amino acids. So these are needed in lower amounts. We have to work all these things out. Half a gram of each of these. 0.12 grams of aspartic acid and glutamic acid. And then you would add vitamins, nicotinamide, thiamine, 0.005 mole peroxidine and zero point 0.5 micrograms of biotin. So what this means is a lot of times vitamins are, we have these additional just sort of trace of each of these things, and unfortunately these have to be added to a cooler type of agar mixture, or they precipitate, which means that they form crystals at the bottom, so they're not in solution, so the bacteria won't have access to it. So we have to be careful on how we make the defined media. A lot of times we do have precipitation problems. In other words, they're not soluble at certain temperatures, and you say, how would you know? And there's a Merck index, MEREK, and I'll provide a link to a Merck index, but that tells you characteristics of just about every chemical under the sun, and you can form a solution with it, and with certain characteristics and that sort of thing. Salts, magnesium sulfate, dipotassium hydroxide, or hydrogen, excuse me, phosphate, 1.25 grams of sodium chloride, and that sort of. Salts typically will cause things to precipitate. So it's usually I'll add this first, and all of these before I'll add vitamins and other chemicals, because the I can manage how soluble, so I would slowly pipette in the vitamin part at a cooler temperature before the agar solidifies, if I'm using an agar, to make sure it doesn't precipitate on me. And also pH is real important. Sometimes I may have to go to 6.5 pH just to make sure that something doesn't precipitate. And see, all those things are a consideration, but for the most part, what's being illustrated here is defined media is that I know every amount of everything that's in that agar, and that is a defined media. Complex media, we can add 27.5 grams of brain heart extract, peptone, 2 grams of glucose, sodium chloride, and disodium hydrogen phosphate is sort of a buffer, and in a liter, and you adjust the pH to 7 and you're good to go. And it's just really quite easy. A lot of times the media that you buy has how many grams, and it usually already contains all the other components, so you just add a certain amount of grams and you're good to go in a liter. But it's complex. We don't really know all the things item by item that's in there, so that's okay. General purpose media, grown as a broad spectrum of microbes. We can have nutrient agars, a good example of general purpose media. Enriched media contains some sort of organic substance as blood. As I mentioned, if I take the blood and I add it to agar that's too hot, it turns to chocolate agar. There are some microorganisms that need that blood component that's actually broken down, like hemoglobin that's been broken down to grow. So, there's uses for it, but blood agars, we usually use it to look at characteristics. Or something needs a special growth factor, or is fastidious. Now, I'm the king of fastidious organisms. Alkalignes faecalis is fastidious. It's kind of hard to grow, especially in minimal media. I mentioned alkalignes before, but you learn to grow these, and you get to know the pathogen or the organism, and so that you can grow it quite nicely if you know all the deals. Here's chocolate agar, by the way. You can see the color. That's why they kind of call it. It looks like chocolate, but it's not. It's just red blood cells, and so that's there. But there's certain organisms that required to grow on there. And here's a blood agar, and it's obviously producing a hemolysin of some kind. You can see that zone around it. You could read a newspaper through there. And there's something about that looks a little disturbing to me. It looks like maybe it's multiple culture here going on, so that I'd have to isolate these and purify it, and just convince myself that it is pure. So that's why we do that. Differential media. Here's a term. I would highlight this, put an X, square, stand on your desk and holler, or whatever. Allow multiple types of organisms to grow, but display visible differences about how they react to that media. So it could be colony size or color, could be media changes its color. So McConkey agar, we're going to talk about that ad nauseum, but McConkey agars I used for gram negatives a lot of times at the anaerobe, the bacteria from your gut. And in that particular case, E. coli, which I've mentioned before, is a bacteria that actually now turns sort of a pink. The colony does. And then the media will precipitate around it. So you see sort of a precipitant around where the colony is growing. That is a two very characteristic of E. coli will do that. Now Salmonella won't, which is a close cousin to E. coli, and it won't do that. There's certain things that can be added. So the reason it turns pink, so the colony actually turns color, is because of the presence of lactose. And E. coli will utilize lactose where Salmonella won't. Various things like that. Or the media color changes. We will use a certain media that will change a color, so it may be a reddish color, and then the bacteria utilizes, let's say you got sodium citrate in it, or we have plates that have this sort of color, and when you grow the organism, it'll turn from a red to a yellow, and so we know that, oh, okay, it's utilizing citrate, and it gives us some information. Or it produces a gas. In other words, it breaks down the sugar and starts creating all these gases. Now it's unabashful. It doesn't say excuse me or anything, but it does produce a gas when it starts to break down certain sugars, and this gas bubbles. Variations often come from chemicals in the media in which the microbes react. So that's a differential. So that's the fun of micro, is that I actually have some of my own media. I came up with a Congo red media. That was something I developed. And a pathogen in E. coli will turn bright red and wrinkle, where a non-pathogenic E. coli will stay white on a Congo red plate. And it's stunning to see it, but that's a differential media. And it tells us a lot about various things. So here's mannitol salts agar, selective differential. It selects for staph because the certain characteristics of some microorganisms, they can grow at high salt because the environments that they typically will grow or will contain a high amount of salt or high osmotic type pressure environment. And this will differentiate staph aureus from other staph. So staph aureus itself has this characteristic. And on a mannitol salts plate, it ferments mannitol and it turns bright yellow. And in this example, so staph are other staph will just be white. So you can see this differential plate over here with staph epidermidis does not turn to plate. The normal color is this sort of kind of salmon color, and in this case the pH is going in the opposite direction. So breaking down a protein and it turns more alkaline, it may be reddish, where staph aureus will turn bright yellow. Look at the difference. So a microbiologist sees these two and we know, oh, okay, staph aureus or epidermidis or some other organism, not always. And E. coli, if you spread that on a plate, it doesn't grow, and it leaves the plate the way it normally is. So we can guarantee that there is, if the sample had some E. coli in there, it won't grow. So it's selective in this case. The selective and differential, the differential is the color changes, and the selective nature is that it grows or doesn't grow. Now I'll say that again, is we use, these are only two that we really focus on in terms of a diagnostic lab. The selective, we don't grow or we do grow, depends on usually salt or something that inhibits. And then the differential nature is like in this we have a indicator, a pH indicator, and it at a acid type of environment turns yellow, or alkaline starts to get red. And so you can see right here in this one example, selective and differential, and how powerful that is. Here's the McConkey agar I mentioned before. It's selective and differential. It's selective because it has bile salts, and it contains a It's part of the gram stain that we use. The gram stain bacteria, it's a actual stain, and that's in there, and it's inhibitory. But E. coli will produce that reddish dark purple color, and you see the precipitant around there. It's like salting out the agar. Very characteristic for E. coli. Where Neisseria gonorrhea won't grow at all, so the plate will either it won't grow or grows very very poorly and doesn't do much, or the Salmonella enteritidis will turn a yellow, and it's very characteristic of this particular organism. It grows, likes the media, and it turns it yellow. But look at the colonies. The colonies stay the normal whitish like color. So big difference between these two. So it helps me identify. Now it may not be Salmonella enteritidis. It could be some other unknown or something like that. But I can at least identify there is a difference. It's not E. coli. This one here is clearly, and then Staph aureus doesn't grow at all, and it leaves the plate pretty much the way we found it. So it does really help us to identify these microorganisms. Sheep blood agar, differential and enriched. So the blood, as I mentioned, we usually pour it in there around 73 degrees or so. Any higher than that, it turns to chocolate agar. But the idea is we see this is a differential but not selective. There is no selective aspect of this sheep blood agar. It's only differential because we look for alpha, beta, and gamma types of situations. So what we have here is beta, usually bad. This is a pathogen of some kind here that's pretty bad, and you can see where it's clearing it. Hemolysin was produced. Here is sort of a darkened area. Is kind of greenish, and that's usually due to hydrogen peroxide production by the organism itself, and it oxidizes the blood or rusts the blood, so it turns that greenish color. Or the gamma, where it does nothing, it's kind of boring. It's just kind of there. It's not really turning the agar one way or another. So, or there's no growth. It looks like it tried to grow, but it just didn't. I don't know. That's probably not the best picture. I would really like not to see anything. I kind of see growth there, but hey, I can't help it. I'm a microbiologist. These are sample slides here. So the types of hemolysis, this will be the last thing I cover for this section. Hemolytic reactions observed on blood agar. We see certain reactions. Now it doesn't have to be sheep. It could be all sorts. It could be cow blood or bovine. It could be ovine. It could be ovine is sheep. It could be canine. Whatever. The blood is blood for the most part, and it's useful for primary identification or preliminary, what I say, a presumptive identification of bacteria, particularly strep. Streptococcus, because of types of hemolysis. Now, strep is a problem, and if it produces a beta-like hemolysis, which is that one that clears, then we know. So alpha is a distinct zone of partial destruction of red blood cells, appears around the colony, accompanied by greenish to brownish discoloration. Streptococcus pneumoniae and other oral strep usually are hemolytic. Beta hemolysis is a clear color zone that you could read the newspaper. It doesn't say that, but you could. Appears around the colonies. They've undergone complete lysis. Streptococcus pyogenes, which you're reading a case study talking about flesh-eating bacteria. Well, here you go. Streptococcus agalacti and several other species of strep are beta hemolytic, beta bad. Many other bacteria besides strep can be beta hemolytic, including Staph aureus, Pseudomonas aeruginosa, and we usually see this organism in burn patients a lot of times, the Pseudomonas. It also produces a green pigment and has a very unique smell, and it would actually show up in bandages as green, sort of greenish color, so you'll know it's Pseudomonas. And the smell of it, you'll never forget it. Listeria monocytogenes, another one. Listeria, a lot of times you get a gastrointestinal disorder, or sometimes it can go in the blood and can be very lethal. Listeria is beta hemolytic. No hemolysis, no apparent hemolytic activity or discoloration is produced. We call this gamma, and so it's useful. Here's a plate that shows all three, and alpha, beta, and gamma. And I like that because it kind of, here's the green, here's the beta, where the zone around where it actually lies. They call this kind of partial lysing due to the hydrogen peroxide, and then nothing happens here with the alpha, beta, and gamma. So I will stop here. This is a lot to digest. If you read through the book and the text, and you will see at least reinforce the concepts of this particular chapter. This is real important. What's nice about this is helping us get ready for the lab part. It's all kind of the same. You will be responsible for this material in the class section, but it also duality of it. This is what I like about this. This is why I waited to do the lab until chapter 2. And because of the kits being late, it follows. So you're not missing much. You're getting the lab material as we go for chapter 2. It's a great thing, and we'll continue on with part 2 of chapter 2 in a little bit.